Welcome to The Explainer. Today, we're taking on a topic that is fundamentally shifting how we view the natural world around us. For centuries, we've looked at biology as this fixed natural science, something we just observe, catalog, and try our absolute best to understand. But what if we completely flipped that perspective? What if biology isn't just a natural science, but a highly programmable engineering discipline? We're talking about synthetic biology, a field that is literally architecting life.
Really think about that for a second. What if we could code cells exactly like we code computers? Keep this overarching operating system metaphor in your mind for our entire time together today. Because in this wild new frontier of science, DNA basically acts as the biological code, and living cells function as the physical hardware. We are actually learning how to write the software of life.
Okay, let's dive into this. Here's our clear roadmap to unpack this incredibly dense, interdisciplinary field today. We'll start with programming life from scratch, look at editing versus building, explore the biological toolkit, see how we're engineering a better world, confront the ethics and limits, and finally, wrap up with the future of biotechnology.
Let's kick things off with Part 1, Programming Life from Scratch, the new operating system of biology.
To really wrap our heads around this, we have to look at how synthetic biology pulls together some incredibly diverse disciplines. We're talking biology, engineering, genetics, and even computer science all mashed up. So the crucial point is right here. It's the science of designing and building biological components, or even entire organisms, totally from scratch to perform specific tasks. We aren't just modifying a thing here or there anymore. By applying rigorous engineering principles, the ultimate goal is making these living biological systems predictable, highly programmable, and incredibly efficient.
Moving right along to Section 2, Editing versus Building Systems. We're going way beyond simple modifications now.
This is where we really need to grasp the massive paradigm shift between older biotechnology techniques and this new frontier. If traditional genetic engineering is like fixing a quick typo in a Word document, synthetic biology is like sitting down and writing a brand new operating system entirely from scratch. Genetic engineering is gene-centered, making relatively simple modifications just to improve existing traits. But synthetic biology? That's system-centered. It designs entire complex biological networks with the goal of creating completely new biological functions that simply didn't exist before.
All right, Section 3, the Biological Toolkit. Engineering principles in the lab. Let's move to and see how this builds. Because if you're going to engineer biology, you absolutely need engineering principles, specifically things like standardization and modularity. And how do scientists actually architect these blueprints in the lab? Well, they use a rigorous design, build, test cycle. First, you design a biological system. Second, you actually build that system physically in the lab. Third, you test its performance. And fourth, you improve the design based on those results. It's a very systematic, iterative loop, exactly how an engineer refines a car engine or a developer debugs an app.
So let's zoom in a bit to the microscopic scale. To build these systems, we need parts, right? DNA parts are the basic genetic elements, things like promoters, genes, terminators, and regulatory sequences. Through chemical synthesis, scientists can actually create artificial DNA sequences. These are literally the foundational lines of code for our biological software, synthesized from scratch and ready to be assembled into larger constructs.
And this brilliantly illustrates how the engineering mindset takes over here. We combine those DNA parts to create genetic circuits. Just like an electronic circuit on a computer's motherboard processes inputs to give you an output, these biological systems do the exact same thing. They switch genes on or off, they control the production of proteins, and they respond directly to environmental signals.
But hey, software needs hardware to run, right? And that brings us to chassis organisms. A chassis organism basically serves as the physical host for these engineered genetic systems. We take our newly written genetic software and boot it up inside common chassis organisms, like E. coli, yeast, actually the scientific name there is Saccharomyces cerevisiae, and even various species of algae. These host organisms act as the actual physical hardware that executes our biological code.
Let's jump to Section 4, Engineering a Better World. Real-world missions.
Armed with this incredible toolkit, which includes computational biology to predict behavior and advanced genome editing tools like CRISPR-Cas9, we start seeing some massive real-world applications. Now, what's really interesting about this slide is how this single technology acts as the ultimate multi-tool for solving vastly different global crises, Look at this. In medicine, it's driving vaccine development and gene therapy. Industrially, it's synthesizing biofuels and bioplastics. In agriculture, we're building disease-resistant crops. And environmentally, we're using it for things like bioremediation and carbon capture. It's one underlying technology revolutionizing 4 completely different industries.
Let's look at a really concrete example to make this stick. The E. coli factory. Imagine you need to manufacture a complex, life-saving pharmaceutical, like human insulin. Back in the day, this was incredibly difficult to source. But today, scientists can design the exact genetic circuit required to produce insulin. Insert that code into the hardware of an E. coli chassis organism, and suddenly, that little microorganism is programmed with the singular mission of pumping out insulin. The cell itself literally becomes the factory.
And it's not just medicine, it's also reshaping how we eat and sustain ourselves. As we try to figure out how to feed a rapidly growing planet, synthetic biology offers sustainable alternatives to traditional manufacturing and farming. We're looking at rapid production of alternative proteins, lab-cultured meat, highly targeted nutritional supplements, specialized food additives, and massively improved crop productivity. It's a fundamental reimagining of our entire food supply chain.
Section 5, Ethics, Risks, and Limits. The responsibilities of creation.
Now, as with any world-altering technology, we have to step back and look objectively at the profound limitations and the really heavy responsibilities of creation here. We are dealing with immense technical complexity and exceptionally high research and development costs, which forces us to ask a highly critical question. What happens when we actually rewrite the rules of life? Handing humanity the keys to life's source code creates a very real moment of tension. There are several specific, urgent concerns we have to navigate. First up is biosafety, ensuring these engineered organisms don't negatively impact our ecosystems or human health. Then there's biosecurity, preventing the deliberate misuse of these highly advanced technologies. Ethically, we're grappling with the creation of artificial life forms, the limits of genetic modification, and those really complex questions about who actually owns synthetic biological products. Because of these very real ecological and ethical hurdles, it is widely recognized across the scientific community that appropriate regulations and rigorous risk assessments are absolutely essential to manage this.
Finally, Section 6, the future of biotechnology. Let's look at where we go from here.
Despite those very real challenges we just discussed, the incredible horizon of synthetic biology at its transformative potential simply cannot be overstated. The future prospects here read almost like science fiction, but they are rapidly becoming science fact. We are moving toward designing entirely artificial cells, developing smart therapeutics, and creating highly personalized medicine tailored to your exact genetic makeup. We're scaling up sustainable biofactories, deploying advanced environmental solutions, and researchers are even working towards creating synthetic organs and tissues. It's mind-blowing.
To sum this all up, synthetic biology is expected to play a major role in shaping the future of biotechnology and sustainable development. Even with those ethical and biosafety challenges that remain, the combination of DNA synthesis, genetic circuits, and computational modeling represents one of the most vital engines for accelerated scientific innovation in the modern era.
I want to leave you with this final provocative thought. We now have the conceptual tools to use cells as hardware and DNA as literal code. So, if you could program biology, what would you build? Think about the vast, world-changing scale of that question. Thank you for joining me on this explainer today, and keep pondering the incredible possibilities of architecting life.